Hello my dear students and welcome for our third class, and topic today's class will be mixed degeneration. Mixed or compositive degeneration are morphological manifestation of impaired metabolism detected both in parenchyma and in stroma of organs and tissues, which occur when the metabolism of complex proteins is disrupted. Endogenous pigments, nuclear proteins, lipoproteins, and minerals. In this slide you may see classification of complex protein. They include nucleoproteins, chromoproteins or pigments, and lipoproteins. Chromoproteins include gematogenous pigment, proteinogenous pigments, and lipidogenous one. Example of these pigments you may see on slide. Classification of mixed degeneration includes metabolism derangement of chromoproteins, nucleoproteins, lipoproteins, and mineral. First of all, we should talking about disorders of endogenous pigments or chromoproteins. So, what is chromoproteins? Chromoproteins is colored substance of viral chemical nature, which are synthesis in our body. Biological function of chromoproteins are transfer of oxygen, realization of respiration, ultraviolet radiation protection, synthesis of bioactive substances, synthesis of secretion, microelement metabolism regulation, and vitamin metabolism regulation. Classification of chromoproteins. According to the structure, we have hemoglobinogenous pigments, proteinogenous or tyrosinogenous pigments, and lipidogenous one. According to the prevalence, they may be general and local. According to the origin, hereditary and acquired. And first of all, we should talking about hemoglobinogenous pigments. They may be normal, which we may find in normal condition. They are ferritin, hemosiderin, and bilirubin. And also in this group we may find some pathological pigment. They are hematidine, group of hematins, and porphyrin. Let's start from hemosiderin. Hemosiderin is an aggregate of ferritin molecules which formed in a cell with excess of iron. For example, with increased hemolysis or with increased supply of oxygenous iron. Macroscopical picture. We may detect it in form of granules of brown color in red cells. Less often extracellular localization of this pathological process of this pigment. The special reaction for hemosiderin and all iron content pigment will be a Pearl's reaction. The detection for iron ions with potassium ferrocyanide and hydrochloric acid. The granules acquired a blue-green color, Prussian blue. So, you should remember that special reaction for iron content pigments will be Pearl's reaction. The second name of Pearl's reaction is Prussian blue or Berlin's blue. Derangement of content of hemosiderin is called hemosiderosis. This condition associated with increasing of this level, and hemosiderosis may be local and general. First of all, we should talking about local hemosiderosis. It occurs with extravascular hemolysis in the foci of hemorrhage. Hemosiderin accumulates in cells surrounding the hemorrhage. In which cell we may find hemosiderin? In macrophages, leukocytes, endothelium, and epithelium. Successive change of pigment formed during the breakdown of hemoglobin leads to a change in the color of hemorrhage. The purple-blue color, hemoglobin, is replaced by green-blue biliverdin, and after it green-yellow hematidine and rusty-brown hemosiderin. An example of local hemosiderin can be brown induration of lungs that occurs during chronic venous stasis in a patient with chronic heart disease. So, we should talk about brown induration of lung because it's our examination slide, and I will ask about it in our models. Brown induration of lungs under the microscope will look like that. You may find some blue-green granules in walls of vacuoles, of alveoles. This slide has special reaction, it's Pearsley reaction. Also in our department we may see slides with standard hematoxylin-iodine reaction, and in these slides we may find that hemosiderin will have brown color, rusty brown color. Microscopic picture of brown induration of lung. The lungs are enlarged, dense, with numerous brownish patches and layers of connective tissue on incision. Microscopically, a large number of cells contain brown pigment and found in the lungs, which can be detected both in stroma of lung and in the lumen of the alveoli and bronchs. The intra-alveolar partition are significantly tinged due to proliferation of connective tissue. Hypoxia is of great importance in the development of hemosiderosis in venous congestion, which is associated with increasing vascular permeability and erythrocyte diapedesis, with the subsequent formation of hemosiderin in cells, in macrophages and alveolar epithelium. In exam, they will ask you about, if you get slide with brown induration of lung, we will ask you why lungs are brown and what does it mean induration of lungs. Induration of lungs associated with development of connective tissue. So, as a result of this process, we may find that lungs will have dense consistent. So, and brown color of lungs will be associated with formation of hemosiderin, which actually have brown color. General hemosiderosis occurs when intravascular hemolysis develops. In disease of blood system, for example, it may be anemia, leukemia, congenital defects of erythrocytes and hemoglobin. In case of poison with hemolytic poisons, in infectional disease, for example, malaria, sepsis, relapse fever, in case of incompatible blood transfusion and rhesus conflict. General hemosiderosis associated with hemosiderin appears initially and predominantly in mononuclear phagocytes of spleen, liver, lymph nodes, and bone marrow, as well as in macrophages structures around other organs in the skin, pancreas, and kidneys. In the future, hemosiderin may appear in the cells of parenchyma, hepatocytes, for example. As pigment accumulates, all organs become brown or rusty color. In most cases, the accumulation of pigment in the organs doesn't lead to dysfunction. Next pigment, bilirubin. Before we start talking about bilirubin disorders, we should remember how it's formed. So, it's formed from heme, from red blood cells, from erythrocytes. So, then hemolysis appear, we may find outcome of gene. After that, heme will have oxygenated and transformed to biliverdin. After biliverdin, it forms non-congestive or uncongested bilirubin. Uncongested bilirubin from blood vessels or spleen transports to liver, and in liver it goes to hepatocytes, and in hepatocytes it transformated from uncongested to congested bilirubin. And congested bilirubin will go to bile, to our intestinal tract, and to our urinary tract, and in our organs. So it's normally formation of bilirubin. In case of bilirubin disorders, we may find disorders of bilirubin metabolism in 3 levels. First of them will be level of blood vessels. If we have find hemolysis, it will lead to increase of uncongested bilirubin and congested bilirubin, because we will have more products of hemolysis in start of this process. Second part of disorders may be localized on a liver, so in hepatocytes. If we have problems with hepatocytes, it will lead to increasing of uncongested bilirubin. And we also may find some problems in bile ducts level. For example, it may be associated with bile concrements and leads to increasing of congestive bilirubin in blood plasma. So, jaundice is an increase of bilirubin content in blood and incretic coloration of skin, sclera, mucous membrane. We have 3 types of jaundice. It may be hemolytic, parenchymatose or hepatic, and mechanical or obstructive type of jaundice. Hemolytic jaundice associated with massive intravascular hemolysis of erythrocytes and increasing generation of congestive and uncongested bilirubin. Causes of this pathological process is blood diseases, for example, anemia, intoxication, infections, transfusion of indifferent group of blood, and rhesus incompatibility. Mechanical jaundice associated with bile outflow obstruction, excretion of congestive bilirubin into the blood. Causes: tumors in the head of pancreas, cholelithiasis, common bile duct compression, portal fissure or portal hepatitis, and metastasis of another malignant tumor. Proteinaceous pigment may include melanin, enterochromaffin cell or granules pigment, and adrenochrome. Function of melanin, the most important function, is protection from ultraviolet radiation, synthesis and deposition of bioactive substances. The most important pigment of this group for you on our exam, on our department, will be melanin. We should talking about it. First of all, we should talking about melanin. Melanin generated in melanosomes of melanocytes. Melanocytes is a cells of neuroectodermal origin. During the tyrosine oxidation into diethylmelanin, the patification of tyrosinase. In the normal condition, melanocytes are located in the basal layer of epidermis. retina, and iris in the pia mater or encephaly. Melanin metabolism disorders may be associated with increasing of melanin quantity, hypermelanosis, or decreasing of it, hypomelanosis. And both of them may be local and general, as well as congenital and acquired. Hypermelanosis, common hypermelanosis. Acquired common hypermelanosis develops in Addison's disease. This disease is associated with lesion of the adrenal glands, with tuberculosis, bilateral tumors or metastasis, amyloidosis, autoimmune damage, and chemochromatosis, and nerve diseases. With the disease, decrease in the function of the adrenal glands, there is an increase in the system of ACTH, which has melanin-stimulating effect. In the skin, the synthesis of melanin is enhanced. It acquires in brown color. It becomes dry and flaky. In the basal layer of epidermis, the content of melanin in melanocytes and keratinocytes increased. Epidermal cells into which melanin is transformed along that process or melanocytes. In derma, melanin is determined in melanophages. Atrophy of the epidermis and hyperkeratosis may develop as a result of this process. Congenital hypermelanosis is observed in xeroderma pigmentosa. It's hereditary disease in which skin sensitivity to ultraviolet rays increases. Manifested by spotted skin pigmentation with the occurrence of hyperkeratosis and edema. It also can lead to development of malignant skin tumor, like cancer and melanoma. Hyperpigmentation, local, of melanin manifested itself from freckless dark brown spots or of lentiga, benign melanocyte lesion like nose, and malignant tumors like melanomas. Hypopigmentation of melanin. It may be common hypopigmentation or albinism. It's associated with hereditary tyrosine deficiency, manifested by white skin, colorless hair, red eyes, and local hypopigmentation of a quite rarely congenital or and called vitiligo or leukoderma. This type, local hypomelanosis, may be development like secondary process after some diseases, some infectional diseases. For example, it may be associated with herpes, with syphilis, and in our lipofuscin. Lipofuscin most often accumulates in the cells of myocardium, liver, skeletal muscles during aging or exhaustion, which is accompanied by the development of brown organ atrophy. Heart becomes small, the amount of fatty tissues under the epicardius is significantly reduced. The vessels become convoluted, the myocardium is dense, brown, microscopical examination characterized by cardiomyocytes are reduced in size, granules of brown pigment, lipofuscin, are visible in the cytoplasm. The liver is significantly reduced, its edge is sharp, the liver tissue is dense, brown in color. Microscopical examination will show liver beams sharply tinged. In the cytoplasm of hepatocytes, we may find numerous brown lipofuscin brown granules. Some hereditary diseases are accompanied by selective lipofuscinosis of internal organs. It's primary lipofuscinosis. It particularly benign hyperbilirubinemia, Dub-Johnson syndrome, is characterized by the development of liver lipofuscinosis. So, right now we should talking about nucleoprotein metabolism disorders. They include gout or podagra, uric acid infarct, and polylithiasis. Gout. It's a chronic disease in most cases associated with a hereditary defect of purine metabolism, primary idiopathic gout. Less often it accompanies with other diseases. For example, it may be associated with nephrocirrhosis, blood diseases, occurs permanently in men after 40 or 50 years. It's characterized by periodic loss of the rights in the small joints of arms and legs, tendons, and peripheral tissues. In response, necrosis and inflammation occurs with development of connective tissue and formation of gouty cones, which deformed the joints. Also, it's very interesting. The gout is a very old disease which we know from a long time, and in 17th or 15th century we have special name for gout. It have name king disease because it associated with big or prevalence meat and red wine in nutrition of organism. Mineral metabolism derangement. The most important for us on our department will be calcium. So we should remember about function of calcium in human organism. The first most important function is skeleton formation. Second is cell membranes permeability, also its excitability of neuromuscular apparatus, blood coagulation, and the regulation of acid-base balance. Derangement of metabolism calcium may have three mechanisms. It will be dystrophic. Calcium may be localized of necrosis and dystrophy phosis. Metastatic, associated with hypercalcemia, and also we may find calcification in kidneys, myocardium, liver, lungs, and stomach. And metabolic, a lung and fatties, aponeurosis in muscles, skin, and in other mucous membranes, for example. And also we should talking today about stone formation. Stone or concrements may localize in gallbladder, kidneys, urinary bladder, salvatic glands, ducts, and staph. Causes of stone formation is salt metabolism disorders, hereditary factors, skeletal congestion or in the ducts of organs, and inflammatory processes. Types of concremation according to chemical composition. In gallstones or cholelithiasis may be cholesterol, pigment, calcific, and mixed. And also we should remember about uralytic concrements. They may be urates, phosphates, oxalates, and mixed one. Stone formation consequence or outcomes of stones. Stone may lead to bed sores, perforation of organs, chronic inflammation, commissures, duct obstruction, hydronephrosis, jaundice, and polylithiasis or urolithiasis. So it's all theoretical part of our today's class. After this, you should do some cases on portal. You may find link by this video. Have a nice day, good luck, and thank you for